Good afternoon, everybody, and welcome to this afternoon's invited master lecture. It's a great honor and pleasure for me to introduce my colleague and friend, Michael Meaney, to you this afternoon. Michael is a professor of medicine in the departments of psychiatry, neurology, and neurosurgery at McGill University, and associate director of the Douglas Hospital Research Center in Montreal. There he is. He is also a senior scientist of the Canadian Institute for Health Research and a senior fellow with the Canadian Institute for Advanced Research. He holds a baccalaureate degree in psychology and biology from Loyola College and an MA in clinical psychology and a PhD in psychobiology from Concordia University. After completing postdoctoral and university research fellowships at Rockefeller and Concordia Universities, respectively, Dr. Meaney came to McGill University in 1985. His many awards include the Patricia Barkas Award, a Distinguished Investigator Award from the Brain and Behavior Research Foundation, being recognized by the Institute of Scientific Information as a most highly cited scientist in the area of neuroscience, and being elected to the Royal Society of Canada. Perhaps most significantly, Michael has also recently been named to the Order of Canada, the country's highest civilian honor, for his contributions to developmental neuroscience. Not bad for a guy whose colleagues tell me scored higher on a risk assessment for ADHD than any of the kids they're now studying. This suggests the possible academic utility of delaying prefrontal myelination until the 6th decade of life. Finally, Michael's work has been nothing short of transformative in helping us understand at a far deeper level what Conrad Waddington referred to in 1942 as epigenesis, quote, the branch of biology which studies the causal interactions between genes and their products, which brings the phenotype into being. In collaboration with his McGill colleagues, Michael's work has produced the first evidence for the epigenetic regulation of the CRH system and of glucocorticoid receptor biology by the character and intensity of maternal caregiving. With its striking binocularity, capturing the twin perspectives of clinical psychology and developmental neuroscience, his work has examined the molecular mechanisms by which maternal care alters gene expression and the subsequent effects on neuronal growth, function, and health. I have sometimes compared the work you will hear about this afternoon to the historic moment at the 1889 meeting of the German Anatomical Society, when the great Spanish neuroanatomist Santiago Ramon y Cajal presented his Golgi-stained micrographs of neurons, proving for the first time that the brain was not a gelatinous syncytium, but rather a network of neuronal cells linked by physical synaptic connections. It was the discovery of these physical, definable connections among neurons that ushered in an entirely new era of studying brain circuitry, the biology of the synapse, and even the psychopharmacology that has transformed modern psychiatry. Similarly, Michael's work has offered us promising new glimpses of the molecular processes, indeed the very physical nexus, whereby genes and environments converge in the determination of both salutary and maladaptive developmental trajectories. Please join me in welcoming Dr. Michael Meaney.
Thank you. Thank you very much. It's an honor to be asked to give this lecture. I, like many members of my family, are shocked by the fact that people would take time out of their day to listen to me. So thank you very much. It's an honor just to have you here. And thank you, Tom, very, very much. Well, we're just a basic Canadian laboratory. And this, by the way, is how you look when you get the Order of Canada. And we're asking and looking at very basic issues. We're looking at development. So when you go back to your basic training in developmental biology, and you read people like Richard Lewontin, who speak of biology as essentially being a process of adaptation. It's an adaptation between the individual and their genome and the context within which development occurs. And it's adaptation to all kinds of forces that we know come to have remarkable impact on phenotype, and that the impact at that particular time in development is then sustained over the lifespan of the individual. And what I'm going to talk to you about today is the degree to which we can think of epigenetic mechanisms as a potential candidate explanation for how it is that this process of adaptation occurring during early development persists in its ability to shape phenotype over the course of the lifespan. And of course, when we talk about the preeminent forces, and this is true regardless of whether you're talking about humans or any other species, those forces are social and economic. They're economic with the respect that every member of every species, to some extent, varies in the degree to which they can access the resources within their own particular niche. So food and nutrients varies across niches, and that influences the phenotype of the developing organism. And of course, we can think of economic issues playing out far more complexly within our own species, but so too is development socially defined by every member of every species, which has at least one parent. So we can come to think of development of any organism as having a social and economic definition. And of course, we can see that when we look at the evidence from social epidemiology in our own species that talks to us about how it is that the context, the social context, the economic context within which development occurs, comes to statistically shape the probability of multiple forms of diseases. And I don't think it'll surprise anyone in this room to understand that family dysfunction in its many manifestations statistically predicts affective disorders, drug abuse, etc. What might surprise some of you is that, in fact, the statistical predictive value of these relationships is no less true when one talks about diabetes, obesity, or heart disease. That health in general and chronic illness are really predicted by these particular types of forces. So the question becomes, of course, how does that occur? Well, a prevailing model within biological psychiatry and other fields is really one that emphasizes the influence of early family life on the development of individual differences in neural and endocrine responses to stress. And I suppose this is best developed when one looks at affective disorders, particularly depression, where we've come to really appreciate, I think, that individual differences in stress reactivity are probably the preeminent endophenotype in mediating the relationship between early family life and the development, at least of this particular disorder. Interestingly, others in the area of cardiovascular disease have made the same argument. Importantly, not only does early childhood experience predict the probability of a disorder, it also comes to predict the severity of the disorder and the ability of the individual to respond to selective treatments. This is a recent paper that was published in the American Journal of Psychiatry, in which the authors provided what is described as the first empirical evidence of any treatment moderator in the treatment of depression. And that, of course, was childhood maltreatment. Interestingly, what was found was that childhood maltreatment predicted the treatment response to pharmacological agents in individuals suffering with depression. It had far less predictive value with respect to psychotherapy, which builds, in fact, upon a report of Charlie Nemiroff and colleagues back in 2000 that showed for individuals with a history of childhood maltreatment, that psychotherapy was statistically more effective than medication alone. So we've come to learn that family history is indeed shaping many aspects, not only the risk for disease, but the way we respond to specific interventions. I think the other feature of this working model, and it is just that, a working model, is its ability to draw bridges, if you will, between those who work in the social sciences and those of us who work in the biological sciences. So, for example, our colleagues in the social sciences would readily point out, as I mentioned, that socioeconomic disparities, poverty, is a major predictor of every single one of these selective outcomes. But what we've also learned, largely in studies done by people in this particular organization, is that poverty comes to afflict the quality of family life, and we can then imagine a cascade that moves forward. Now, this is a working model, but I say one of its great virtues is the degree to which it brings together people collecting evidence at the level of social and psychological outcomes, as well as those looking at biology. Well, here's another component to this that, as coming from a background in biology, I can't ignore. Just to revert to this particular slide, the basic theme in this slide is really that exposure of the parent-child unit to adversity in early life ultimately comes to influence the phenotypes, the outcomes, the developmental outcomes in the offspring. And that is a theme that is by no means unique to human beings. So here's a slide that summarizes many, many years of work in evolutionary biology. These are individual scientists working with fairly simple models by our standards. They're working with plants, insects, birds, occasionally mammals, but for the most part, insects. And what is uncovered is that when the parental organism is exposed to adversity, nutrient deprivation, violence, infection, and predation, that it alters in some way a signal from the parent to the offspring that then changes forevermore the phenotypes of the offspring. Now, the nature of that parental signal varies. Obviously, if we're talking about plants or insects, it's going to be of a fairly simple and no doubt a chemical form. But the major feature of this is very much coherent with what we see in human psychology. I'll just give you an example of this. These are skinks. And I guess I'm the only person at this meeting showing a photograph of skinks, right? It's probably safe. Well, these are cute, small reptiles, and they're heavily preyed upon by snakes. If the skinks that are most successfully preyed upon are those that are smaller, they have shorter tails, and they're less behaviorally reactive to the cues of snakes, so they don't escape as effectively, and to put it bluntly, they're easier to swallow. If at any point in time over the course of her history, the mother was exposed simply to the scent of a predatory snake, then her offspring are born larger, they have longer tails, and they're behaviorally more responsive to the cues of a snake. Now, if you think about it, that's essentially what we're talking about, right? Exposure of the maternal organism to adversity is in some way represented in the development of the offspring. And so I think what we're talking about, however, and I greatly appreciate the fact that this is going to be infinitely more complex in the human context, there's something very fundamental about this in the context of biology. So here's the message. Or skink. And in fact, actually, we do see evidence of this. Of course, this is just the summary of a presentation that was made this morning from our MAVEN group. Is simply looking at the degree to which maternal adversity, that is the mother's developmental history, predicts negative emotionality in the child, even independent statistically of her own mental health. And of course, there are multiple examples of this. So in certain ways, the adversity of the parent is transmitted to the offspring. And the question then becomes, how is all of this occurring? And so here's where we get to what is the question that drives our laboratory. It's essentially trying to understand what is the basis for these enduring or programming-like effects whereby environmental signals act over various phases of development and associate with stable changes in the way in which the genome operates, transcription, and thus enduring changes and effects on complex phenotypes. So here's the bottom line. What I hope to present to you is some preliminary evidence that suggests that this type of plasticity, this parental influence on the phenotype of the offspring, has in part a basis that is actually apparent at the level of the DNA. And that, of course, refers to epigenetics. Epigenetics is broadly defined, really, as any functional change in the genome that does not involve a change of sequence. And it's really that phrase that historically separates us from Jean-Baptiste Lamarck. But we're talking really about is the biochemical mechanisms that regulate gene transcription. So what I'm going to do is walk you through a little bit of what we look at. Now, I want to tell you that this isn't really all that hard. If it was, we couldn't do it. What we're going to do is to talk a little bit about epigenetics, which has a long history, right? People, by the way, anybody who wants to come in, I'm used to people walking out of my lectures, so it's a treat to have somebody come in and walk in. So if you'd like to, I'm not easily disturbed. Unfortunately. So epigenetics has this long history, and it essentially has been this mysterious black box. Whenever an organism was exposed to a particular experience and then later on showed a change in phenotype, people would say, well, it's epigenetic. Well, as it turns out, epigenetics has a very real scientific and biochemical basis to it. And it starts out with the DNA itself. Now, here's one of the real puzzles if you're looking at gene transcription. What you're looking at right now is, in fact, crystallographic representation of how DNA is normally packaged. It's usually packaged in a nucleosome, tightly wrapped around a core of histone proteins. Okay, so here's 146, 147 base pairs of DNA, and it's wrapped tightly around these histone proteins. And that configuration is maintained by things like electrostatic bonds. Histones are positive, DNA is negative. So it's a tight configuration, and the reason why, if you're asking yourself, you should care about such things, is that when it's tightly wrapped around the histone cores, the DNA cannot physically interact with a class of proteins known as transcription factors. So it tends to be silent. The reason for that is that the activation of DNA, DNA in itself is inert. It needs to be activated. It's activated by cellular proteins called transcription factors. They bind to regulatory sites in the DNA, and that turns on the activity of the gene. When this configuration is closed like this, the transcription factor cannot interact with the DNA. Therefore, the DNA is silent. You want something to happen from this site, you've got to physically remodel it. And the site at which this remodeling occurs are these tails, these amino acid tails that protrude outside the nucleosome. The tails belong to the histones. And what you do is you actually make some subtle biochemical modifications to those tails, and in doing so, you open up chromatin. The most common modification is you simply take a little acetyl group, you slap it onto the histone tail, it partially neutralizes the positive charge of the histones, opens up chromatin, and now the transcription factors combine. So whenever genes are active, two things are usually occurring. You're opening up chromatin, and then the transcription factor binds to the DNA. So you've remodeled chromatin, and that's happening all the time. But these are very transient events. Acetylation of histones, it comes on and off. It's a matter of seconds or minutes. Haven't quite figured out how long it occurs, but it's very transient. So it isn't the kind of stuff that's going to explain how exposure to a particular experience at one time can have an enduring effect on gene transcription. But there are modifications that last longer, and they incur methylation, that is the addition of methyl groups, in part onto histones themselves. And people like Eric Nestler at Mount Sinai have shown that methylation of histones can have long-term effects on gene expression. But our work is even more fundamental. We're actually dealing not with modifications to the histones, we're dealing with modifications to the DNA itself, and in particular, the classic epigenetic mark of DNA methylation. So this is just simply taking a methyl group and adding it on, or not, to particular regions of the DNA. And in mammals, the only nucleotide subject to methylation is cytosine. So you look across the genome, you're going to find lots of cytosines. Some are methylated, some are not. What we want to do is to understand why that's important and how that process may be regulated by environmental signals. Well, the first part, why is it important? It's important because when the DNA in these regulatory regions, these regions right here that activate the transcription of a gene, when they carry a methylation signal, they attract a class of proteins known as methylated DNA binding proteins. They then bring a complex of proteins to the affair, and the major element of this complex is an enzyme known as histone deacetylase. And what it does is exactly what its name suggests. It kicks the acetyl groups off the tails and prevents acetylation. And if you can't acetylate, then you can't open up chromatin, and it remains silent. How's that? See, I told you it's not hard. Biochemists could never do it. Well, that's the process. That's it, really. And the interesting thing about DNA methylation is it can be enduring. It can last for long periods, in presumably the lifetime of the individual. And it allows us to ask some really fundamental questions about biology. So every one of us, right, embodies, literally, the paradox in genotype-phenotype relations. That is, every one of us produces about 220 different cell types, but we do it from a common DNA template. Every cell in your body has the same nuclear DNA, yet you can produce cells that are as different as liver cells, bone cells, and brain cells. And the way you do that is by epigenetically silencing certain regions. So you want to make a cell a brain cell, you want to specialize it to do the kind of things hopefully we're doing right now. Then you leave those genes on, but you turn off genes that are associated with metabolizing lipids and glucose. That's the job of the liver. Liver cells, well, genes that are involved in playing chess or doing mathematics are turned off. Those involved in lipolysis and glucose regulation are left on. So you're specializing the cell by epigenetically regulating the genome. So developmental biology has obviously embraced epigenetics. Then it gets even further interesting because what you're looking at right here is one of the classic features of development in psychology, and that is how can monozygotic twins who share the same nuclear DNA differ so widely? We know that discordance rates amongst such twins for diseases, even diseases that are widely heritable, is much less than 50%. So how do you do that? How do you create such variance in phenotype? And it may not surprise you to know that the mother of these two perfectly identical twins doesn't have a lot of trouble telling them apart, even when they have the good grace to leave their shirts on. So these processes we know are involved in creating divergence, multiple phenotypes from a common genotype. And our question is really simple. Could the same mechanisms then be involved in explaining the enduring influence of environmental events? So we want to get at this, and we propose a fairly basic hypothesis. We refer to this as the environmental epigenetics hypothesis. It suggests that environmental events activate intracellular signaling pathways, which we know occurs, of course, and that those pathways and their activation can then functionally remodel certain areas of the genome with respect to these epigenetic signals. And that once remodeled, these epigenetic signals can then carry forward, if you will, the signature associated with that particular experience. If you will, in a way, we think of epigenetics as being an experiential registry that's imposed on the DNA. That's the hypothesis. And here's how we test it. We look at a non-human species, of course. We're looking, in this case, at long Evans hooded rats. And what you're looking at here is a mother who is interacting with one of her multiple offsprings. And you see this licking behavior. And mothers vary, believe it or not. Mother rats vary quite reliably. There are mothers who lick their pups very frequently and other mothers who lick much less frequently. And our question is, can this stable individual variation in the interaction between mothers and pups, can it give rise to epigenetic marks that then can be related meaningfully to changes in gene expression and to broader measures of phenotype? So our first goal is to really ask the question, can we relate parental care and variations in parental care to these epigenetic marks? So we're looking at this licking behavior, which we knew from scientists who had preceded us was a behavior of profound relevance for the development of the animal. So people like Saul Shanbert and Cindy Kuhn had shown that mom's licking activates the release of growth hormone. And that, of course, stimulates growth. Gig Levine had shown that the same licking suppresses pituitary adrenal activity, sparing the animal exposure to the highly catabolic glucocorticoids during a period of time when, amongst other things, brain development is occurring. So this, in essence, social event is regulating the immediate physiology of the pup. And our question then becomes, can it be something that has a longer and more enduring signature? So the typical type of experiments we do, we spend 6 to 8 hours per day watching mothers interact with their pups, sort of a perfect activity for Canadian winters, which are long enough to permit lots of data. And then what you find, as I mentioned, is that there's normal variation. If you look at licking as a trait, as we'll do towards the end of the talk, it's normally distributed, and there are mothers who lick their pups consistently 2 to 3 times as frequently as do other mothers. And what we do then is just what anybody in developmental psychology would do. We simply take the offspring of these animals and we follow them through to adulthood. And we find that they differ very reliably in terms of both their behavioral as well as their endocrine response to stress, which you'll see, They differ in neural development. Areas like the ventral and dorsal hippocampus, the prefrontal cortex, are structurally modified by the mother's licking. It underpins the differences in certain forms of learning and memory. They're different metabolically. By mid-age, a great percentage of the offspring of low-licking mothers are obese and insulin resistant. And they differ in terms of reproductive development, at least do the females. The females reared by low-licking mothers, and this will resonate with many of you, go through early puberty. So there's an awful lot of coordinated changes occurring across phenotypes. And of course, then what becomes critical in terms of using a non-human model is to be able to functionally relate the variation in postnatal maternal care to these developmental outcomes. We do this simply by doing cross-fostering studies in which we take the biological offspring of low-licking mothers and foster them onto high-licking mothers and vice versa. And when we do so, we can in part or completely reverse each of these expected differences in phenotype. So we know that mother's behavior then is driving, at least in part, these particular phenotypic outcomes. So our question then is that of drilling down on a particular phenotype, and to do that we've looked particularly at the stress response. And of course this will be well known to most of you. What you're simply looking at is the pituitary adrenal stress response, which is activated by a master stress hormone called corticotropin releasing factor. It is released by the hypothalamus in response to any form of stress one can think of, activates pituitary ACTH and the release of glucocorticoids from the adrenal. Now, where this system becomes under even further brain regulation is when you start looking at what it is that regulates CRF, that master stress signal itself. And what happens is that the glucocorticoids, cortisol in primates, corticosteroid in rodents, acts on glucocorticoid receptors in the hippocampus as well as other brain structures. And when it interacts with that receptor, it sends an inhibitory signal to the hypothalamus that dampens the synthesis of CRF. And so the activation of glucocorticoid receptors is commensurate with inhibition of CRF and a more modest response to stress. And here's the bottom line for the adult male or female offspring of high and low licking grooming mothers. Animals that are reared by high licking mothers show increased glucocorticoid receptor gene expression. They make more of these glucocorticoid receptors in the hippocampus. Ergo, they have tighter control, more efficient control over CRF, and thus more modest ACTH and corticosteroid responses to stress. By contrast, the adult animals reared by low-licking mothers show reduced glucocorticoid receptor expression, less efficient regulation of CRF, and thus exaggerated ACTH and corticosteroid responses to stress. And here's what it simply looks like when you take the adult animals reared by these mothers and expose them to an acute stress. You see that at both the level of pituitary ACTH or adrenal corticosteroid, there's an increased response in the adult offspring of low compared to high-licking mothers. And we can completely eliminate this effect if we use either pharmacological or genetic procedures to disrupt glucocorticoid signaling in the hippocampus. So we know that it is the glucocorticoid action on the hippocampus mediated by the glucocorticoid receptor that's responsible for this difference. And that takes us to a point where we can identify a particular gene. We can also, of course, do the cross-fostering study, and cross-fostering in this case completely reverses the phenotype. So animals born to highs but reared by lows show reduced glucocorticoid receptor expression and greater responses to stress. And conversely, animals born to lows but reared by highs show the more modest response that is associated with increased hippocampal glucocorticoid receptor expression. The other dimension to this that's rather interesting is the precision of this, because if you look across the brain, or indeed in other areas of the body, you find that glucocorticoid receptor gene expression is in fact undifferentiated as a function of maternal care. The animals don't differ. It's only within the hippocampus and small regions of the prefrontal cortex in which one sees this maternal effect. The other way you can do this, and this is some studies we did in collaboration with Marion Joels now at Utrecht, is to actually become a little more obsessive about it, if that's at all possible. And in this case, what we did was, rather than scoring the licking behavior of the mother, we scored the licking towards individual pups and then followed those pups into adulthood and were able to statistically correlate the degree of licking to glucocorticoid receptor expression in adulthood in the hippocampus. So it is the licking behavior, you'll see further evidence of this as we move along, that's driving this particular difference. So now we get to the point we can start to look at what we always wanted to look at, is how can a variation in maternal care, which is physiologically relevant for the stress response, how does it produce a sustained effect on transcription? Because of course we wean our animals at 22 days of age. After 3 weeks of being with their mothers, they move off, they don't write, they don't call. They are liberated from parental influences thereafterwards. So this is a sustained effect on gene expression. So we've got to drill down to the relevant molecule, and of course that's the gene that produces the glucocorticoid receptor, and what you're looking at is a schema of what that gene looks like. In pink are segments of DNA called exons that collectively produce the glucocorticoid receptor protein. In blue are regions, and this is an area that was cloned by myself and our lab and Jonathan Seckle's lab in Edinburgh. This is a region that contains regulatory sequences. They don't contribute to protein, but each one of these 12 sequences can determine the level of activity of that part of the gene that does produce protein. So they're regulatory sequences, we call them promoters. So our question then becomes, well, which one of these regulatory sequences is influenced by maternal care and produces the difference in the glucocorticoid receptor in the hippocampus? And the answer is it's in large part this exon 1,7 sequence. So it's a promoter that's largely very active within neurons. And as you can see from this graph, it's far more active, expressed at a much higher level in the adult offspring of high compared to low-licking mothers. So this is the promoter that is largely responsible for the increase in glucocorticoid receptor expression that we see in the offspring of high compared to low-licking mothers. Okay, now what we want to do is sequence it. We want to know individually what comprises this region. And when we sequence this promoter, we find that it's got a region that can bind a specific transcription factor called NGFIA. And that's relevant because NGFIA is the transcription factor that turns on the glucocorticoid receptor gene. So literally what you've identified here is the location on the DNA where that transcription factor is interacting with the DNA to turn on the glucocorticoid receptor gene. Okay, now what we want to do, having looked at this particular sequence, is to start looking at its epigenetic basis. Now, if you'll notice in this sequence, you've got two cytosines, one here on the left and another on the right. Either of these cytosines, and particularly those that are neighbored by a guanine, that is C and G, they're real candidates for methylation. Okay, so what we want to do then is to characterize whether or not these sites are methylated and whether or not the methylation status is influenced by maternal care. So we go in, we take the hippocampus out from the adult offspring of high and low licking and grooming mothers, and we prepare the tissue to analyze the methylation state. And what we find is that this one cytosine right here is differentially methylated as a function of maternal care. The one on the right generally tends to carry the methyl mark regardless of whether the animal was reared by a high or low licking grooming mother. The one on the left carries the methylation mark generally if the animal was reared by a low licking mother, but not if it was reared by a high licking mother. So here's an epigenetic signature, if you will, that associates, statistically at least, with maternal care. So then we go into our cross-fostering study, and we ask, is maternal care really driving this difference? And here's what we find. You see that, again, in animals born to and reared by low-licking mothers, you generally find the methylation signal at that site. If the animal is born to and reared by a high, you don't. If the animal is born to a low but reared by a high, you see less methylation than if the animal is born to a high and reared by a low. So it's maternal care that appears to be driving this particular difference. So then we ask, is it functional? Is it really something that could underscore the differences in glucocorticoid receptor gene transcription? And I mentioned to you earlier that when sites, particularly sites in regulatory sequences such as promoters, carry this methyl mark, they can attract these repressor complexes that include histone deacetylation. That, if you will, prevents the acetylation of that particular region and thus tends to associate with silencing. So we went through and we did a whole series of assays, and what you find is that when you pull out that region of the DNA from the hippocampus, that the region that carries the methylation and thus is occupied by these histone deacetylases is less acetylated than the same region in the offspring of high-licking mothers that doesn't carry the methyl mark. This area is more acetylated, hence it's open. Well, if it's open, it should bind more NGFIA, and it does. And if it binds more NGFIA, then it should be more active, and it is. So here you have the link. Now, the real critical question is, can you then go into this system and reverse this process? So what we do is we take adult animals reared by high or low licking and grooming mothers, and directly into the hippocampus we infuse a compound that inhibits these HDACs. And when we do that, we completely reverse every one of these effects. Okay? So it is the methylation and the histone deacetylation that's associated with that. But you turn around and say, well, that's fine for gene transcription, but we're really interested in stress responses. Well, here's the stress responses from that particular experiment. Here's the corticosterone response to stress in animals that are born to and reared by low-licking mothers and infused with the vehicle, and you see the exaggerated corticosterone release from the adrenal. If the animals were reared by highs, in the blue and red circles, they show the more modest corticoid response to stress. And importantly, animals reared by lows, but in which we reversed this epigenetic signature, the response is the same as it is in the highs. So reversing the consequences associated with methylation is sufficient, if you will, to reverse the maternal effect on the pituitary adrenal response to stress. So it does appear as if the differential methylation at this site is associated with changes in glucocorticoid receptor expression in the hippocampus and differences then in the magnitude of the response of the animals to stress. And we've done, oh goodness knows, about 12 years of work trying to figure out how all of this occurs. Essentially what happens, interested in the details, is that maternal licking, as people like Myron Hoffer and Saul Schamberg and others showed many years ago, it activates the sympathetic system. And it's interesting, of course, that licking occurs at the time the mother's nursing the pups. So what mom's licking is doing is to activate metabolism during that particular period, and part of that involves thyroid hormones. Thyroid hormones regulate the release of serotonin in hippocampal neurons, and it is here that you activate this intracellular pathway that actively remodels this particular site. And the key element for this, and we've done in vitro and in vivo studies to show this, the key element is the simple binding of the transcription factor to that region of the DNA. That's what triggers the change in the epigenetic state. And we can go and show that, in fact, it is the tactile stimulation of the mother that's critical. For example, Ian Hellstrom plays mom. He simply takes pups and he brushes them with a horsehair brush to mimic the tactile stimulation of the mother, and that's enough to increase the binding of this NGFIA to that particular region of the DNA. And this and other experiments, I think, give us some confidence that it is the tactile stimulation derived from maternal care that drives this particular effect. Well, we then get to a question which is inevitable, of course, and one that's teasing you all along, is whether or not anything comparable might be happening within our own species. So this is obviously a fairly complex set of experiments, and it's one that is by no means free of complication as far as interpretation are concerned, and I trust each one of you can think well of that. So I'm going to just tell you about what were the resources that we had in terms of being able to address this question. So the group at McGill, including Gustavo Turecki, Moishe Ziff, myself, etc., we're working with brain tissue from what's known as the Quebec Suicide Brain Bank, which is run by Gustavo. Quebec Suicide Brain Bank is, as the name suggests, a repository for brain tissue from individuals who are victims of suicide versus individuals who have died involuntarily. And we can go in and excise the hippocampus. That's not a complication. Then also we bring to strength something that Gustavo has developed, which is a validated forensic phenotyping. This is an interview process, as I say, validated, in which Gustavo's team is interviewing the family of the victims to gauge the level of current or previous psychopathology as well as developmental history. And we can look at the same region of the genome which has been characterized subsequently. And this is the organization of the human glucocorticoid receptor gene. It is very similar to that of the rat. And what you'll see is that this exon 1F region here is the ortholog of that exon 1-7 sequence that we studied in the rat. So what we do is we've got hippocampal tissue, and now what we simply do is to look at gene expression, measures of gene expression. We do simply PCR analysis, and what you find is that in those individuals who are victims of suicide, there's a decrease in the total amount of glucocorticoid receptor messenger RNA. There is also a decrease in the level of activity of this exon 1F promoter. And then what we did was to see whether or not we could relate this particular difference in gene expression to the differences in the mental health of the individuals. And so we correlated it to the level of depression and drug abuse, and we found nothing. And our sample sizes were pretty good. We had 36 samples, and we were reliable enough, I think. We looked at other forms of psychopathology, but the sample size was too small to draw any inferences. And then what we started to do was to look at developmental history. And in these particular samples, we had well-validated measures of maltreatment. And as it turns out, about half of the 24 samples from the suicide group were individuals with characterized and well-documented cases of childhood maltreatment. And it was only amongst those individuals in which one saw the difference. So suicide individuals free from a history or a known history of childhood maltreatment did not differ from controls. It was uniquely viewed amongst the abused. And that was true not only for the message that produces the protein, it was also true for measures of this exon 1F promoter. And then, as you can well imagine, what we did was to look at the methylation levels, and we found the methylation levels mapped on to the developmental history. So there was decreased expression of the glucocorticoid receptor gene, which, like the rat, was associated with an increase in the methylation of this promoter, this exon 1F promoter. And the region that was differentially methylated lay right next door to a region that binds NGFIA. And what happens then is that the increase in methylation decreases the ability of NGFIA to interact with the site and turn on the glucocorticoid receptor gene. Now, this is a finding that fits very nicely, as it turns out, with previous studies that have been published in the literature. Work from people like Michael DeBellis and the group at Emory, led by Christine Heim, have shown that childhood maltreatment associates with an increase in both CRF levels in cerebrospinal fluid, as well as greater autonomic and pituitary adrenal responses to stress. So somewhat like the rat, individuals who are victims of maltreatment show decreased glucocorticoid receptor expression and greater pituitary adrenal responses to stress. Now, interestingly, of course, our studies are conducted with hippocampal brain tissue, which surprisingly few Canadians are willing to donate to us. And so it becomes then interesting in the extreme to note that two other laboratories have essentially replicated this particular profile that is showing that early childhood adversity associates with differences in methylation, and they were looking at exactly the same sequences. In fact, they used the same primers as we did. So they have essentially replicated this, and what they're doing in this case is to look at leukocytes, and I think this was done with red blood cells, which of course people are a little more free to give up. And what it means is that potentially one can then trace these particular marks in cells that are accessible. And that brings it from a finding that is of some biological importance into one that might be clinically useful. And I'd like to come back to that particular point in very soon, in fact. Right. So here's where, and I'm throwing this out to you as a matter of thought. And believe me, this is by no means at this point in time a conviction, but it's where we think, it might be interesting to take the whole issue of epigenetics. When we look at the type of predictors that we have of psychopathology, they could be socioeconomic misfortune, they could be birth weight, gestational age. We've got many predictors of poor mental health outcomes, none of which, unfortunately, are all that good. So, I mean, even if one looks neurologically at children who are born premature, about half of them do extremely well without the benefit of any intervention whatsoever. Similarly, when we look at socioeconomic disadvantage, we can find, yes, there are individuals who grow up in poverty, but especially where there's family cohesion, they do particularly well. And the other big problem we have is, statistically at least, is that the majority of individuals, and this is a slide ripped off from the late Clyde Hertzman, is that many individuals are found within the middle class, right, in areas that escape our particular predictors of depression. So the point is, we essentially need better predictors of vulnerability for the sake of our treatment studies. We need better predictors of treatment outcomes and of who might be stratified to particular treatments. And so these reports we find interesting because they suggest to us that we might be able to see the epigenetic signatures in individuals by looking not at the brain necessarily, but at cells and tissues that we can access and thus use clinically. So we tried to look at this, and this is just some preliminary evidence, and so please, this is very preliminary. We're working our way through this, and the statistical pitfalls here are almost innumerable. But let me tell you a little bit about what we've been doing. This is from our longitudinal maternal adversity and neurodevelopment study in Montreal and also in Hamilton. What we've done is to take individuals who were classified using the strange situation test as either secure versus insecure at 18 months of age. And then at 6 to 8 years of age, we go in and we take, in this case, not blood, but epithelial cells from the cheek swabs, and from which we can extract an enormous amount of DNA. We then take that DNA and we run it against an array. This is done in collaboration with Mike Kobar at University of British Columbia, and we're looking at an array that interrogates 488,000 individual sites along the DNA. And we're asking, can we see meaningful variation in the methylation of any of these sites as a function of insecure versus secure attachment? Now keep in mind, about 60 to 70% of all the methylation across the genome is invariant. It's never going to vary, right? So if you're ever interpreting data along these lines, we've got tons of viral DNA, right? A lot of it. That has to be methylated, otherwise we're going to look pretty funny. So you've got to methylate viral DNA. You have to methylate other particular regions that you want to be active, et cetera, or don't want to be active. So every cell in the body is going to share about the same 70%. It's only about 1 to 3% of all the sequences that are likely to vary as a function of their epigenetic state, which interestingly enough is still a bit more than the number of sequences that vary across our DNA, right? I mean, only 1% of DNA sequences show variation in humans. Hasn't discouraged genetics, nor should it. So with that in mind, what we found is about 600 or so probes, individual CPGs, that differ by more than 5% and are statistically reliable. And some of them are really interesting, the kind of things you get all excited about when you do these sorts of things that have absolutely no statistical validity at all, but otherwise are great fun. They're genes that are involved in long-term potentiation or synaptic plasticity. And then there are, of course, genes that you will recognize. And so let me tell you a little bit more about what we did then. So we say, okay, we've got all of these probes and genes. Let's use gene ontology. Let's ask what these genes might have in common with respect to predicting particular disease outcomes. And here's our number one hit. At the very top of the list, the genes that collect or cluster together are genes that are associated with ADHD. And you can go into gene networks and pull out these ADHD gene databases. You can argue with some of them to be sure, but for the most part, they give you some idea that these genes do tend to cluster. So we look at these particular genes and we find some hot ones that we know differ on our arrays and are associated with ADHD. Unfortunately, the bioinformatician didn't know the DBH stands for dopamine beta hydroxylase, or this guy being yellow too. But we've certainly got some things like COMT, catechol methyltransferase, and DRD4 that make the list. Then we've got, this is a child development project. We've got things like the strength and difficulty questionnaire, and we correlate the individual CPGs of certain selective on these genes with the inattention scale on the SDQ, and you can find some correlations. Now, some of you are out there thinking, yeah, but what's the chance finding here? You're right. Okay, so this is all very preliminary. But then we get to something interesting and highly unexpected. We found that methylation on one gene correlates with methylation on another gene in many cases. What you're looking at here is the methylation of a major variant on COMT with that on DRD4. And these genes are on completely different chromosomes, so it's not that they're neighbors or anything. Then we went ahead and we started to do what's called a cluster analysis. Now, apart from looking really good as a t-shirt, what this analysis, at least that's my thought, what this analysis tells you is that if you look at all those 123 probes, those individual sites that were associated with ADHD, the majority of them actually are co-regulated. They come together as a cluster. And that, as was unexpected, and the clustering here is actually pretty dense. You see right in here 30 individual probes where the average correlation is 0.8, right? Not bad. So what we're coming to is a point where we think, okay, possibly what we might be able to do then is to talk not about individual CPGs, which I think is a bit naive, but we might be able to characterize people using clusters, the way geneticists use haplotypes, clusters of variants rather than single variants. So this is just, if you will, it's nothing more than simply giving you a walkthrough of the way in which we might be able to ultimately translate this particular work. So, some thoughts. How many things for time? We're good? Oh, I got more slides. Shamelessly enough. Some thoughts, and please, I mean, this is work in progress, right? Childhood maltreatment clearly does predict the risk and potentially even the form of affective illness, as well as many other types of diseases. It tends to distinguish, as my colleague Robert Leviton at University of Toronto points out, atypical from melancholic depression. So it may be allowing us to stratify beyond what we normally think of in terms of our global diagnostic outcomes. This may be overstated, but our developmental predictors right now alone are not really sufficient. And I was talking this morning to some people from the great work of Craig Ramey and others in the Abecedarian project, in which he had cited a graph in looking at the gain, and I think it was IQ, but I could be corrected, but it was a gain in children from disadvantaged backgrounds as a function of their mother's education. And the issue there, of course, is that the kids who came from mothers who had not experienced a high level of education showed a great treatment gain, whereas mothers who had had some college education or beyond, the treatment gain was very small. So here, the real risk, of course, is that what you do when you combine all these individuals across a single group is that you underestimate the gain that could be achieved if you had more accurately stratified the kids who truly were at the greatest level of risk. And so, I mean, are our interventions, were these kids any worse off from being in the project? No. But the problem is in assessing the treatment effect and the effect size, that if we don't have more accurate markers and predictors, we may be underestimating our intervention sizes. We need objective reporters of developmental history, and we need them who occur earlier on in life, even prior to the point at which individuals can report on their own developmental history. And it's not clear by any means, and I think there are many people in this room who would echo this concern, that the more extreme forms of adversity are necessarily the only predictors of morbidity and treatment outcome. There are studies, however modest one may think of the measure. The parental bonding index is a remarkably good predictor of the risk for depression. It's not clear that by simply appealing to these extreme forms, we can capture that vulnerable group. And it may be that vulnerability then is emerging as a function of underlying, if you will, genetic or herited vulnerability, as well as the environmental interactions. And if so, epigenetics might be an interesting way of stratifying those populations. All of this, as I mentioned to you, is really thoughts, right? Long way from convictions. So I want to talk to you about one last project, one last theme here that we've explored that I hope to some extent intellectually closes the circle. I've talked to you about how parental care, I think, can drive, and we've got others, people like Tiwan Zhang in the laboratory have shown that everything I talked to you about for the glucocorticoid receptor works for genes that are involved in regulating glutamatergic or GABAergic neurotransmission. So GR is kind of our poster boy, if you will, but there's many other genes that fall into the same profile. Okay, so when we go back and are informed, as we should be, by the epidemiological evidence. And in this case, this is just a graph ripped off from Ken Kendler's many wonderful articles on the pathways that lead to depression. You're struck by the fact that there really are cascading series of experiences that occur here. And so, for example, childhood maltreatment associates with poor outcomes with respect to social support. Peer networks among such kids generally tend to be of poorer quality. They tend to be of lower education and lower education achievement. There are personality variables such as neuroticism that are influenced by this. And so there are these cascading pathways of influence that occur. It isn't simply that something happens and then down the road you get sick. It's all the stuff that happens in between. And I know I'm preaching to the converted here. But it does enlighten us with respect to the way we need to think about things like methylation. We need to, for example, ask the question, do these epigenetic marks follow this same particular type of trajectory? Are they modifiable by subsequent experience? And so that's a very rudimentary question that was addressed in an experiment done by Dr. Tiwan Zhang in our group. And so what he did was to take animals that had been reared by high or low licking and grooming mothers, and he weaned them as we normally do. He then put them under standard or enriched housing conditions. And they remained within those conditions from 22 to 70 days of age. And for those of you who haven't worked with rodents, enrichment is a remarkably profound environmental procedure. It changes almost every dimension of the phenotype of the animals. And then what he did to make sure that whatever epigenetic signatures were associated with enrichment were enduring was he put all the animals into standard housing conditions until they achieved 90 days of age, well past sexual maturity. We then used an array that we had actually designed ourselves that sampled across 2.1 million regions of the DNA, the whole genome, 27,000 genes in the rat. And we asked, to what extent is the variation in the methylation across this region associated with maternal care compared to environmental enrichment? And what we found, and this is just a principal component analysis of that question, is that enrichment actually accounted for about twice as much of the variability in methylation as did variation in maternal care. Now, of course, there's an interesting feature to this in that enrichment's more recent, right, than is maternal care. All kinds of things to think about, but it clearly suggests that the epigenome is responsive to these downstream changes at the level of enrichment and methylation. So, okay. Broad changes that are associated with parental care. They involve, as I mentioned, changes in neural development, another day, another talk. Metabolism, same thing, but they also involve influences through the reproductive development of the females. Part, as I mentioned to you, is associated with pubertal development. But another developmental outcome associated with reproduction, influenced by maternal care, is maternal care itself. So interestingly enough, the great female rat nightmare comes true. They become their mothers. So if you take animals that are born to females that are born to high-licking mothers and you breed them, on the whole, they show a substantially greater level of licking towards their offspring than do females that are reared by low-licking mothers. Frances Champagne, who's documented this, Frances is now a professor with her own lab at Columbia, and Frances showed the correlation between the levels of licking of the mother and of her female daughter. What she also went on to show was that this is correlated across generations, so one can not only statistically correlate the mother's licking with that of her female offspring, but even the grandmother's licking to the female offspring. So that, I think, fits it to some extent within the work summarized by others, by Jay Belsky and others in humans, that there is this evidence for transgenerational transmission of variation in maternal care. The second feature of the model that I think gives it some fidelity with respect to the human condition is that it's also, like human parenting, subject to the current environmental conditions. So this was another experiment done by Frances in which she showed the impact of chronic stress on the quality of maternal care in the female rats. She took high and low licking and grooming mothers, and she bred them and characterized them. And then for the second litter, one half of each group was exposed to chronic stress during the last part of gestation. And what she found was that when she looked at the licking to the second litter, that mothers who had been characterized as high-licking mothers and were unstressed were again high-licking mothers. Low-licking mothers remained low-licking mothers, but those who had previously been high-licking mothers but were chronically stressed now were phenotypically indistinguishable from low-licking mothers. So chronic stress can then modulate this entire phenotype in a way that I think is consistent with what we understand in the human condition. So we started to look at the developmental origins of this, and the first thing, of course, we wanted to do was to verify the cross-fostering, that it really was the postnatal maternal care that was driving the maternal care of the female offspring, and the cross-fostering studies, I think, were unequivocal in this. This was work done by Darlene Francis as well as Francis Champagne. Here in green are the groups that were reared by high-licking mothers. The critical group was that was born to lows but reared by highs. In this gold color, you have the animals that were reared by low-licking mothers, the critical group being those that were born to highs but reared by lows. So cross-fostering completely reverses the phenotype. And interestingly, of course, then if you take the female offspring of these animals here, who were previously characterized as high-licking mothers but had behaved over this litter as a low-licking mother, and you follow their offspring, what you find is that amongst the controls, you see the expected transgenerational transmission. Animals raised by high-licking mothers show more licking to their offspring than do animals raised by low-licking mothers. But if the mother was stressed and behaved thus as a low-licking mother, her female offspring behaved to their offspring as does a low-licking mother. We've then gone on to piggyback onto some of the great work in neuroendocrinology, beginning with Jay Rosenblatt, going through Michael Newman's work, Cord Pedersen and Bob Bridges and many others who have precisely and beautifully documented the neuroendocrine systems responsible for activating maternal care in the rat. What you find is, essentially, that as Jay Rosenblatt described, the trigger for initiating maternal care in the rat is the expression of the estrogen receptor in a brain region known as the medial perioptic area. Once activated, the estrogen receptor turns on the oxytocin system in the medial perioptic area. Oxytocin neurons project down to the dopamine neurons of the ventral tegmental area, and they stimulate the release of dopamine in the nucleus accumbens, and that's what activates licking and grooming. And the critical trigger for this is already different in the female offspring of high and low licking grooming mothers. The female offspring of high licking mothers show more estrogen receptor in the medial perioptic area than do the female offspring of low licking mothers. Consequently, they also show differences in oxytocin and the oxytocin system. And the oxytocin receptor and its expression in the medial perioptic area strongly predicts the variation in maternal licking and grooming. So the estrogen receptor in the highs is expressed at a greater level. It sensitizes the oxytocin system. It turns on more oxytocin, and that associates with an increase in dopamine. So what you're looking at here are actually signals, dopamine signals, in the nucleus accumbens, and in the pink bar indicates a period of licking and grooming. So you can see that low-licking mothers show an increase in the dopamine signal in the nucleus accumbens when they're licking their pups, but in the high-licking mothers, that's much more exaggerated. There's a much greater release of dopamine in the nucleus accumbens, and that correlates with the frequency of pup licking and grooming. So the bottom line is essentially it's the activation of estrogen and estrogen drive over oxytocin systems that causes the release of dopamine, and you can eliminate the difference in the dopamine signal in the nucleus accumbens by blocking the effect of oxytocin at the dopamine neurons. So this is simply the level of dopamine in high-licking mothers infused with saline, block oxytocin, and they release dopamine at no greater amount than do the normal low-licking mothers. So now we've got a single gene, we think, the estrogen receptor, which activates a cascade that produces this difference in licking and grooming. And that difference in the estrogen receptor is also reversed with cross-fostering. Here is the expression of the estrogen receptor in the medial perioptic area of animals reared by high versus low-licking mothers. If the animal is born to a high but reared by a low, it shows very modest estrogen receptor expression. If it was born to a low and reared by a high, it shows increased estrogen receptor expression. Good. So now we think we've got a gene that's triggering this difference in maternal care. Let's look at the DNA that regulates it. And you know what's coming, right? I'm a one-trick pony. It's going to be epigenetic. Here's the area of the estrogen receptor gene, this 1B sequence that drives, it's the regulatory sequence. It drives the transcription of the estrogen receptor. And sure enough, it's differentially methylated as a function of maternal care. So in black, you see the level of methylation at all of the individual 13 cytosines or CpG dinucleotides across this exon 1B promoter. And you can see that clustered across almost the entire promoter, there's increased methylation in animals reared by lows compared to animals reared by highs. And we've gone on to show that that too is associated with differences in the ability of transcription factors to occupy the site and turn it on. But let's cut to the chase. The estrogen receptor is differentially expressed. It appears to drive these differences in maternal care, and the difference in the estrogen transcription seems to be epigenetically modified. Is the differential expression of the estrogen receptor what is sustaining the transgenerational transmission of these variations in maternal care? Is it really the activation of the estrogen receptor with its downstream effects on oxytocin and dopamine that is the trigger for this? So let me tell you about an experiment that we did. We've got a way that we can genetically modify the expression of the estrogen receptor in that critical brain region, the medial perioptic area. We take what's called a short inhibitory RNA sequence. It binds to the normal messenger RNA that produces the estrogen receptor, and it essentially inactivates it. So when you infuse this, you couple it to a lentivirus so it infects all these cells, and it's stable. It becomes incorporated into the cell, and it reduces the ability of the cell to produce a functional estrogen receptor protein. So essentially what we can do is to eliminate this difference in the estrogen receptor. But we're only doing it in one brain region, right? The rest of the body is just fine. We've just put it right directly into the medial perioptic area. So here's the experiment. And this is a cruel thing to do after you've been patient enough for all this time, but I promise this is it. We take animals, females, and we breed them and characterize them as high or low licking and grooming mothers. Then we take their female offspring, and we know that under normal circumstances, these animals, all things being equal, will generally be high licking mothers, and these will generally be low licking mothers. We take one half of the offspring and we infuse them with this shRNA. We neutralize or diminish their ability to produce the estrogen receptor alpha. That's this particular manipulation right here. And then what we do is we ask, okay, we've eliminated the difference in the estrogen receptor. Does that eliminate the difference in maternal licking? And it does. So I think quite predictably, eliminating estrogen receptor alpha differences in the medial perioptic area eliminates the difference in maternal licking. Now we get to what is really the show. Now we want to look at the next generation. No manipulation here. No estrogen receptors, no viral vectors, nothing. All we're asking is, can we now see the impact of this on the next generation? And the answer is yes, you can. You see it not only in terms of estrogen receptor expression, they express the estrogen receptor as did their mother, you also see it in terms of licking and grooming. So the epigenetic modification of the estrogen receptor and its expression is what's sustaining the difference transgenerationally in maternal licking and grooming. So as promised, summaries. To date, some evidence in favor of an environmental epigenetics hypothesis suggesting that variations in profoundly important manipulations, and again, it's lovely to speak to an audience that understands that maternal care is something that normally is involved in regulating phenotypic development. It is associated with sustained differences in the epigenome. That is true of the rat and may be true within our own species, remaining to be determined. It thus also seems to serve as a potential mechanism, and the link here is, of course, maternal behavior, right? That's what's serving to transmit these states across generations, which, if nothing else, simply goes to confirm something that's painfully obvious to everyone at this meeting, is that parenting is a profoundly important target for epigenetic modification, because it is what is sustaining these transgenerational patterns over time through epigenetic processes, we think. Thank you ever so much. Thank you.
This session is being recorded, so if we could pass this mic to people asking a question, it would be very helpful in recording the entire question.
Or I could repeat it.
Or you can repeat
I could repeat it. As opposed to giving an answer. Yes. Yeah, that's okay. The question is, could these epigenetic modifications fine-tune statistical relationships between genotype and phenotype? That would be exactly the expectation. And environment. Yes, absolutely. So there are, and I'm blanking on the paper that was published that is in fact a proof of principle on this line. It's done with the serotonin transporter gene. And it showed exactly that particular effect, that indeed you are fine-tuning these relationships. You could even, at the point of going from actual data to speculation, imagine how it then opens up or closes windows of plasticity accordingly. Absolutely. Yes?
To what extent is the methylation a graded response as opposed to a binary?
Okay, so what extent is the methylation a graded versus a binary response? In truth, it's a binary response. The problem is we measure it as a graded response. And here's one of the great weaknesses we have, particularly when we work with brain tissue. We take a whole range of cells, we grind them up, we extract DNA, and we look at the methylation signal and present it as if it's graded, when in fact in individual cells, depending on the number of alleles, it's 0 or 1. And that's a huge problem. Until we can get to the point of being able to characterize methylation marks in individual cells, we're not going to be able to do this. We can get slightly better resolution when we sort cells by particular phenotypes, and we do that, but you're absolutely right. In the real world, when we think of how this is working, it's binary. It works collectively as a graded, but it's binary. It's either methylated or it's not. Yeah, that's a really interesting question, because then, okay, I've given you the classic cell biology where it's binary, it's 0 or 1, but cells work in networks. And so it may well be that if we can define an individual network, it may be that it's across that network that matters. It could even be that, in fact, and this I think is almost inevitably going to be the case, that we need to move from looking at individual sites to looking at waves of methylation. I spared you a few slides where, in fact, we've looked at this. Methylation tends to not be here and then not here. It tends to be clustered around the genome. And so it may be the size of that cluster at that particular region that's effective as well. Yeah, it's a lot to be determined in terms of some very precise questions about how it operates in terms of being able to subsequently interpret the data. But you're absolutely right.
At a functional level, they have sort of threshold with maternal behavior. Maybe a bad mother or a good mother can build sort of.
Yeah, we've looked at I highlight the high-low comparison. In most of the studies, we've actually got data from mothers that fall in the intermediate phase. Most of the measures are generally linear, and that's true of the methylation signals that we've looked at thus far, but not all of them by any means. And in fact, fear behavior is one of the interesting exceptions to that. In terms of the way you respond with fearfulness on many particular measures, if you're a mid or a high, it doesn't matter. It's the lows that are different, and there are other measures where it's not. So you're absolutely right. We need to keep. It's all the more reason to understand what's the biology underlying the maternal care effect in particular tissues. But you're quite right. How that works across these thresholds, we don't know. And even to the point, embarrassingly enough, of knowing how the methylation properties across regions of the genome function in concert, we're not clear about yet either. And the clustering across areas takes it to another level. Sorry, was there a question there?
Yeah, I was curious. You seem to have some parallels between the licking and child maltreatment, and I was just wondering. The licking seems to be more of a positive thing. I don't know if it activates parasympathetic nervous system in the rats, and maltreatment seems to be mostly traumatic and doing something rather than absence of something. So I'm wondering how far that analog goes.
Yeah, the question is, how could we compare across high-low licking mothers and versus maltreatment? I think I'd leave Dante to perhaps speculate on the maltreatment and its underlying properties and what it particularly predicts. That's not my area of expertise. I think the best point is you're absolutely right. We're not trying to. Now, what we did do, and in fact, this was led by Matt Suderman and Moishe Ziff at McGill, who took the data from the humans and the data from the rat and superimposed it on top of each other. And to be honest, there's a lot of limitations in that because the genomes don't line up perfectly well. But it's not hard to find areas that are vulnerable to parental influences. The nature of those parental influences in the two models, I think, is just a totally different thing. To be honest with you, I would step back and say, at very best, we've suggested variations in parent-child interactions can influence epigenetics in both species. Beyond that, the comparisons, I think, are a bit strained.
Is kind of correlative of the absence of some parental mechanism.
That's a really, really interesting question, is that, of course, what captures us when we think clinically about childhood maltreatment is the abusive but there's much more packaged into that, including the absence of what might be more positive forms. And again, I think that's a great discussion, but it's one that weighs, unfortunately, beyond our data set. Yes?
When you group your offspring by high and low living mothers, do you categorize them by how much the individuals got lived on a general basis, or is it if the mother picks one pup a lot, everybody goes into that?
Yeah, it's the question is, how do we categorize our animals? Normally, we throw everybody into the same basket based on the mother's behavior, but we have, at the behest of our dear colleague Dr. Joels in the Netherlands, become more sophisticated and done experiments where we've looked at individual pups. We've done it as a way of being able to look at within litter variation, and the one may look at within family variation. And it maps on very nicely. So for example, I think I showed you the glucocorticoid receptor. That works with synaptic plasticity. It works with BDNF and other measures that we've looked at as well. But you're right, the statistical model is limited by the fact that we're clustering those pups based on the mother's behavior as opposed to the behavior to individual pups. That level of resolution would probably give us, we would assume, slightly stronger effects. Oh, sorry.
Talking about epigenetic mechanisms underlying oxytocin receptor expression. Just wondering if you are aware of epigenetic mechanisms for the production of oxytocin itself, particularly because we've got so many studies coming out which are nasal oxytocin effects on.
Yes, no, I think that's absolutely true. The question pertains, do we have information, or is there information on the epigenetic regulation of the oxytocin gene itself? Very little at this particular point in time. I know there are people very enthusiastically looking and characterizing the methylation state of the receptor, much less so the gene. One of the reasons that might be true, and I would defer anybody interested in that topic to Larry Young's work at Emory, which is the best in terms of looking at variations in oxytocin gene expression, is that oxytocin is very short-lived. And so what Larry's been focusing on is not so much oxytocin, but the signal that drives oxytocin, which is a melanocortin receptor. And actually, it turns out that when he uses compounds, drugs that activate that indirectly signaling oxytocin, he gets much bigger effects than oxytocin itself. But no, I don't. At this point in time, and I know we've got a very close colleague, the oxytocin gene was actually cloned by Hans Zing, who's the chairman of our Department of Pharmacology and Therapeutics, and Hans is all over us to look at the oxytocin. We will do it, but to the best of my knowledge, no, it hasn't been looked at yet. Oddly. Tom, you want to sort? I'll go 1, 2, and 3, okay? Just for...
Yeah, is it comparably easy to reverse the methylation, or is it pretty much a one-way process?
No, it's actually biochemically, and all I can do is speak about a few studies, but more so the biochemical mechanisms. It was initially thought, and there was a certain hesitancy in accepting some of the data that we have now published, and for quite some time ago, because it was thought that methylation is maintained by carbon-carbon bonds, hence it's unbreakable. Now, I don't know when you did chemistry, but when I did chemistry, Carbon-carbon bonds weren't unbreakable. They're high-energy bonds. They're not easily broken, but they're not unbreakable. So we proceeded accordingly. It's not easily reversed, and it appears that the biological mechanisms by which one does reverse it, that is, take a methylated state, turn it into an unmethylated state, involve DNA repair systems. Just to give you a little detail, the DNA repair system recognizes a methylated cytosine as a thymidine and wants to turn it back into a cytosine, so it really goes through a uracil to cytosine conversion. That's a pretty convoluted step, and it's a high-energy step, but there's a lot of evidence now that it is and can be done. And Howard Seder, who's been one of the great pioneers in the study of epigenetics at the Weizmann Institute in Israel, documents, classic cases of overdevelopment of demethylation. There's wonderful cases of it occurring. The processes by which it occurs have really only recently come to light. They're a little bit convoluted. It's clear that methylation isn't going to be quickly or easily reversed necessarily, but it's highly reversible, yes. Yes? Oh, thank you. That's good. Yeah, it's a great question. The question is, are there critical periods for this? And my colleagues, I know, have given a lot of thought to this. The problem with the critical period thing is that it's a phenomena that doesn't necessarily map on easily to any biological system. So, for example, I could turn to you and say that maternal licking or maternal care has its greatest effects on methylation at this particular point in time. But is that a function of the organisms having lost the ability to respond to methylation? Or does it now respond to some other age-appropriate event that activates the same intracellular pathways with the same vigor and is just as responsive? So it doesn't respond to mom, but it responds to something that's more age-appropriate. As a biologist looking at the cellular machinery, I would favor the latter, because we can take cells in culture. Remember I mentioned, I'm challenging you on this, there'll be a quiz. Remember I mentioned NGFIA's binding to that site is critical for producing the epigenetic state. Well, we can take a construct, methylate the hell out of it, put it in culture, and then drive it with NGFIA at any point in time, and it demethylates. We think that the mechanisms there are sufficient at any point in time, at least the cell. Now, the critical period may then derive from the properties of the organism and its response to the environment, but not the cell per se. Hope that makes sense. Yes? Sorry, the use of epithelial cells? Yeah, to what extent would I expect there to be the methylation signature on epithelial cells? Sounds a bit wonky, doesn't it? Well, if you're as old as Tom is, then that was a cheap shot, eh? I'm going to pay for that. But you will think back to biological psychiatry in the 1980s and 1990s, when people were actually looking at relevant receptors, serotonin, noradrenaline receptors, in platelet cells, in blood cells, lymphocytes, etc., and finding things that actually came to anticipate very nicely what we've now learned from positron emission tomography. Should the epithelial cells or platelets or lymphocytes reflect these processes? It sounds somewhat counterintuitive until you start to think that we're actually thinking about these type of early experience events as triggering global responses in stress hormones, in immunological signaling, in cytokines, which we know are released during periods of stress and adversity, and that it might not be beyond, I'm speculating, but it might not be beyond the pale to say that exposure to adversity excites neural pathways, but also peripheral signals that potentially have effects on the epigenome across many different cell types. That's kind of the way we're coming to think about why it might be, but I think it's one of those instances where the proof is in the pudding. Can you actually predict? Now, we do have studies, by the way. Gustavo, Mike Kobar, to some extent ourselves, etc., are taking the post-mortem tissue and blood in human. Jonathan Mills at University College London has done some of this work. We're in non-human primate. We're comparing epithelial cells to brain and just trying to find those actual correlations. But I think the bottom line is, can you actually use it to predict particular outcomes? I mean, we can, except, yeah, we can. We've done it in blood in rats, absolutely. But it's just more, it's actually not that hard if you have the access to the human brain to get blood from autopsies and correlate the two. And in primate, you can do it in a way that's more anatomically informative than it would be in the rat. Sorry, am I missing? Did I prompt? I go 1 and then 2. Yes?
Michael, what about individual differences in susceptibility to methylation?
Fantastic question. Individual differences in susceptibility to methylation. No, I didn't put the slide in because it's a bit preliminary, but there's one study that's been done on the serotonin transporter, and we have data on the DRD4. So, for example, what I showed you as being an effective attachment on the DRD4 gene is one of the genes that was listed. That's modulated by the variant VNTR, the 7 versus non-7. So what happens then is if you have the VNTR, then you are sensitive to attachment. If you don't, at the level of methylation, if you don't, it makes no difference whatsoever. I'm so glad you raised that point, because epigenetic signatures will be a function of gene environment and genome, and in part just simply because genetic variations will be determining the nature of the cellular response to the relevant environmental event, but in part it'll be because the sequence itself will have properties that make it more or less vulnerable. It absolutely will occur as a function of both. Yeah, that's absolutely great. Thank you.
Last question.
Last question. There was somebody. Yes, hi.
How much do we know about how the internal looking learning behavior ends up increasing the FIA?
Yeah, how much do we know? A lot, a whole lot, actually. So what happens, well, since it's the last question, and since we worked so hard for this slide. I mean, 12 years of work. So I think, to go back and show you this slide one more time is not, if you will, overly indulgent. Assuming I can get there. And there it is. This slide right here. Yes. So here's what happens. Mother's licking, at the down end of all of this, mom's licking is directly activating NGFIA, increasing the protein, and it's binding to that region, and that's what alters the epigenetic event. But what's critical is that if you take the animals as adults, their production of NGFIA is exactly the same. So it initiates the event, but isn't involved in sustaining it. But it's absolutely critical, and as I was talking about before, we can just take cells, hippocampal cells, and overexpress NGFIA, just using genetic tools, produce exactly the same effect at this particular level. So yeah, that's the nexus around which it's occurring. And it isn't just NGFIA, it's bringing a whole bunch of bodies to the party that seem to be critical as well, but that's really it. Mom really is initiating these cascades that are remodeling the epigenome. Thank you ever so much.
Thank you very much.
Thank you. Thank you.